Good afternoon, everybody, and like hopefully all the speakers, I will also start out by thanking the organizers for inviting me to this amazing meeting. I've already learned a lot, and if the first 1.5 days are anything to predict the next 2 days on, I'll probably learn a lot more, and I've already enjoyed amazing talks and stimulating discussions. So what I will talk about is an all-atom structure of a HIV capsid. And let me start out with some general remarks. Most of you know that there are several methodologies around with which you can get all-atom structures of biological molecules. Everybody knows about crystallography. There were a number of talks here that represented structures that were solved by crystallography. Those structures are, as the name will tell you, solved in the solid state on a crystal. Solution NMR works in solution. That means we do not have the necessity to crystallize our proteins. So in that sense, we have one advantage. You also saw about some of these big advances that have been made in Cryome recently. And if you have entities, molecular structures or assemblies that do not lend themselves to crystallography, very frequently Cryome will be able to give you resolution structures that are now approaching atomic resolution. You also heard that there is another methodology that, in my view, right now is really rapidly advancing structural work, and that is solid-state NMR. And the big advantage of solid-state NMR is that you can work on amorphous systems and irregular aggregates. So anything that associates and does not form order is very well suited to be studied by solid-state NMR. But if you really want to solve a biological problem and get an answer that is meaningful, it is best to really combine all of those methodologies, because only if we do that, then we can understand a system in detail and maybe exploit it either for drug design or to design new experiments that will probe function. Now, in addition to all of the structural techniques, we should not forget that we can get an awful lot of information from other methodologies. And I very much believe that we have to integrate a lot of experimental techniques, structural as well as cell biology, biochemistry, spectroscopy, and put all of this together with in vivo studies, because what in the end we want to do, we want to understand what a particular macromolecule does. And really what we want to understand it at is at the level of the atoms. If you want to understand a chemical reaction, you need to know which atoms are involved and what they do. If you want to understand interactions and how proteins, let's say, bind to a ligand or to another protein or to a nucleic acid, you really want to know what types of atoms are doing the job. So in Pittsburgh, we have a center that's called the Pittsburgh Center for HIV Protein Interactions. And we were very lucky that around 6 years ago, we got funding from the National Institutes of General Medical Sciences to study some aspects of the HIV life cycle in terms of trying to get structural information. Our particular center has as a scientific focus immediate post-entry events. These are the events after the virus attaches to the cell, the viral and cellular membrane fuse, then the contents of the virion gets released into the cytoplasm, and then a lot of magic happens. There are events that are called uncoating, and then reverse transcription starts. Then the PIC, the pre-integration complex, gets formed. That docks onto the nuclear pore. This gets then into the nucleus, and then in the next step, the reverse transcribed viral genome gets integrated into the host genome. Now, what is here depicted at this empty sea of light blue, in reality, as you all well know, is not such an empty light blue sea. It's a very complicated, hostile environment. And the virus has to make sure that it can traverse this hostile environment and do the functions that are necessary to get into the host chromosome without being first obliterated by all of the host cell defense mechanisms. So the mechanism, or one of the most important entities that play a role there, is this particular entity here, and that's called the capsid core. The capsid core encloses the viral genome, an RNA genome, and then interacts with some of the components of the cell till in the end we have this pre-integration complex at the nuclear pore. Now, for those of you who are not familiar with HIV, HIV has become sort of the poster child for a virus now. There are more studies on HIV than on any virus around, and the HIV virus life cycle is frequently used as a model system. And I'm not going through all of that. I only want to give you a few glimpses that I think are important about HIV. First of all, the genome is an RNA genome, and it is made up of 9,000 nucleotides. That's 1/1,000,000th of your genome. So it's ridiculously small. It encodes 9 genes. It's really a paltry number. And those 9 genes encode 15 proteins. That's it. So this very simple, stupid virus can wreak havoc with your immune system and basically shut it down. So lots of studies have been gone on. The one thing I'm only depicting here is one of the proteins that is important for making this conical capsid core. And like most proteins of HIV, it is produced as a polyprotein. It's called GAG. At the N-terminus, we have called a protein that's called matrix. Then comes the capsid protein and the nuclear capsid protein. And then there are a few peptide appendages around. The capsid protein makes this conical shape. And again, for those of you who heard about viruses throughout this meeting, in contrast to some of the viruses where you heard structural work on, the HIV virus is an enveloped virus. So we actually have a cell membrane. In the cell membrane, the major receptor that interacts with the cellular receptor sits. It's called the glycoprotein GP120. Then on the inside of the mature virion, you have this matrix protein that lines the membrane. Here you have this conical shape, the capsid, made out of the capsid protein. Inside depicted here are the 2 RNA strands that make the genome with 2 RTs in there, and there's a handful of other cellular proteins that usually gets packaged. Now, in contrast to this beautiful artist's rendering of a virion as shown here, if you look at virions in real life, either when they bud here from a cell surface or here isolated viral particles, they don't look nearly as nice and beautiful and symmetric as what's depicted there. And even if you look inside and try to locate the capsid core, you'll see that occasionally you see something that looks like this conical shape, but then you have little stubby things in there, sometimes rod, sometimes some things look really a little bit messy. And you can make those purified cores and look at them, and here again, it's probably a minority that has this beautiful conical shape. The rest looks all different. So it's a very pleiotropic arrangement, these capsid cores. The one measurement I want you to remember is that the average core diameter is roughly 44 nanometers, and that will come handy in later to remember that number. Now, the entire capsid here is made out of a single protein. a fairly small protein, only 231 amino acids, made out of 2 domains, the N-terminal and the C-terminal domain, and both domains are helical. And there have been quite a number of crystal structures of those domains around over the years, for the last 25 years. There was only one structure of a full-length protein that existed. That was from Steve Kusak's lab, and he crystallized the protein in presence of an FAB fragment and found that it was in this head-to-tail arrangement in a dimer. Now, people knew very early on when they looked at cores that on the outside of the cores were only N-terminal domains. So this particular arrangement cannot be the arrangement how the protein is arranged in the native capsid core. We also recently got a structure in Pittsburgh, and again, it's one of these inhibited head-to-tail dimers, now without an FAB. All the different structures have different arrangements, for instance, when it comes to the protein-protein interfaces. And in particular, if you look at the C-terminal domains, they're always dimers. But the way the dimer is arranged in the crystals varies quite a lot, and I've only shown 2 here of the CTD interfaces. Either the helices are almost parallel, or the crossing angles are all different. Although the monomeric unit is basically identical, the way the monomers are arranged in the dimer is quite different. And even in the head-to-tail dimer of the NTD-CTD in the full-length protein, you can see that helix 9, which is really the helix that is at the interface in the C-terminal domain, is also the helix that makes the interaction there. So the question arises, is this plasticity that you see in the intermolecular interaction in these dimers, is this important for capsid function and assembly? The first information that it may be came from a very early cryo-EM study where Barbie Gansner-Perillas took in vitro assembled capsid, flattened it on a grid, got a cryo-EM mat, and tried to fit in the crystal structures that were out in the PDB. And she could fit the N-terminal domain in very easily. It was a good fit. Any of the C-terminal domains out there, the dimers didn't fit. She had to take the dimer apart and fit in the individual units to make a fit. And that really was the start for us to look at what the C-terminal domain looks like in solution, because maybe it was different from what out there in the crystallographic database. And I'm not going to show you a lot of NMR, but I'll just show you a few spectra to illustrate where NMR can be extremely powerful. For instance, lots of us in the structural world use the divide and conquer approach. So you make a domain and you hope what you see in that domain is what the protein looks like in the entire protein. If you're an NMR spectroscopist, you would label your protein with N15. You record a HSQC spectrum, which basically is a fingerprint of what the structure looks like, because it only looks at amide correlations between amide protons and amide nitrogen, and everyone gives you a little dot there. And then you take the spectrum of the domain, you superimpose them, and if the domain spectrum has all of the dots in the same place as they are in the full-length protein, you can be fairly confident that the structure is identical in the isolated domain to the one that is in the full-length protein. And this is definitely true for the N-terminal domain of the capsid protein. If you do this with the C-terminal domain, there was a little bit of a complication because not all of them seem to be fitting of the magenta spots onto the black ones. And I've blown a few things up here on the next slide, so you can see some magenta spots are on top of black ones, but others are not. And what we found out was that the C-terminal domain in solution exists as a concentration-dependent equilibrium between a monomer and a dimer. And you can see this very easily here from a superposition of a low concentration spectrum that's shown in cyan and a high concentration spectrum in red. And some residues only exhibit resonances here in the red spectrum, and I've circled them. Now you can take just one of the monomeric units out of any of the crystallographic structures, and then you map, since we have assignments, those resonances onto the structure, and you immediately map out the dimer interface that way. And not surprisingly, helix 9 is in that interface. You can also use it to determine the dimerization constant from the concentration dependence of the spectrum, and it's probably a very expensive way of doing that by NMR. Now, another very powerful NMR experiment when you want to check whether a structure is different in, let's say, a crystal or in solution, is to record residual dipolar couplings. You don't need to know anything about how it's done. The only thing you need to remember is that a residual dipolar coupling gives you information about an interproton bond vector, the orientation of that. And let's say we're looking at a nitrogen proton vector, so you can determine how this vector is arranged in space with respect to the molecule's anisotropy axis, or for all intents and purposes, the magnetic field. And then you can take a structure out of the PDB and calculate those residual dipolar couplings and plot them against the ones you measure in solution. Now, if the structure is identical or very similar to what's in the crystal, you should get a straight line at the diagonal. As you can see, for 4 that I've shown here, there is not very much resemblance. So we knew that the structure we had was not similar or identical to the dimeric structures that were out in the crystallographic database. So we had to determine the structure by NMR, which we did, or in this case, in Jabourne in the lab did that. Here is the structure, one monomeric unit shown in pink, helix 9 in magenta on one unit, the gray other monomeric unit, helix 9 in cyan. And if there are NMR people in the audience, this is a well-defined structure. This is as good as we can do them. And as you see here, helix 9, we have a nice interdigitation of hydrophobic residues that make up that interface, at the center of which sits, let's say, tryptophan 184. We can also be very confident about mapping out the details of an intermolecular interface by NMR. And this you do by taking a sample that is N15C13 labeled, a sample that is at natural abundance. You mix it up, and then you carry out an experiment that is called a C13 N15 filtered and then heteronuclear edited experiment, NOSI experiment. In these types of experiments, you only see interactions between a proton that's attached to a heteronucleus and a proton that's attached to a natural abundance nucleus. So you very cleanly can map out intermolecular interfaces. And you can see how nice, clean these spectra are. Here I show you interactions around this tryptophan 184 side chain to valine 181, methionine 185, isoleucine 15, and so on. So this was expected. This is helix 9. We also saw another intermolecular interaction, and that was sort of a surprise, because we saw that tyrosine 145 sits at an intermolecular interface. Now, as a structural biologist, if you see something like that, you always immediately think this must be important. But just because it's structurally important does not mean it's functionally important. So you have to go and test whether it is functionally important, and that's exactly what we did. We made 2 mutants. We changed tyrosine 145 to an alanine and to a phenylalanine. And then in vivo, we tested core stability, infectivity, and RT activity. These are parameters that will tell you whether a virus behaves like a bona fide, a wild-type virus. And as you can see from the data, look here at the green bars. These are pretty dead viruses. They're non-infectious. And just for the chemists in the audience, I always like to point out the only thing we've done, we changed one atom in there, namely got rid of that oxygen. Now, it was sort of a surprise because in the PDBs, where there's a structure of the N-terminal domain that was solved by Chris Hill quite a number of years ago, he has tyrosine 145 in the N-terminal domain. It's actually the capping residue of the last helix in the N-terminal domain. We have it in the C-terminal domain at an interface. So where is tyrosine 145? Actually, it can switch conformations. And if I show you the overall full-length structure here, you can see that it sits at the hinge region between the N-terminal and the C-terminal domain. And depending in what environment it is in the N-terminus, it just caps off that helix, so it makes an intramolecular interaction. If you have it in the C-terminal domain, either the isolated domain or the full-length one, it makes an intermolecular interaction. Now, when we did our CTD NMR dimer structure, we had a slightly different construct than what was crystallized. And most people don't even worry about that. But you always say, oh, maybe it's conditions, or maybe it's a slightly different construct. That is the difference in the dimeric arrangement. So we worked on a protein that starts at position 144, and Chris Hill crystallized the protein that starts at position 150. And people would say 6 amino acids doesn't make any difference. Yes, it does. If we made that construct, we looked at the spectrum, and the first thing we immediately noticed that the dimerization constant was different. If you make it shorter, it's actually a much weaker dimer by a factor of 5. And what we also noticed was that if you look at the spectrum at 25 degrees and just look at those resonances that arise from a tryptophan and epsilon proton, there were 2 resonances, and that's true for lots of others too. So there are actually 2 forms in this particular construct here. We did our structure at 25 degrees for the longer construct, and we could not see another form. However, if we go to low temperature, 7 degrees, we see a tiny bit of a second form there. So we can pick up even different conformers for the dimer by NMR. What was interesting to see is that these 2 dimers behave differently, and in the long dimer, we can actually switch conformers by pH. If you work at net neutral pH, and obviously we did our structure neutral pH, we have this one conformer we did the structure of. If you go down to pH 2, you see that the second conformer comes up, and we can really titrate that. And it's basically just the protonation of one side chain, the glutamate 175 side chain. If you look at the shorter construct, you obviously don't see the pH effect. This side chain is there. It's not that it's not there, but you don't see this pH effect, and you have basically always a 50-50 mixture of those 2 conformers there. And maybe it's the D2 conformer that likes to crystallize and not the major one in solution. So it's always good to do both, maybe crystallography and NMR. What we really want to look at is the overall conical arrangement of the capsid, and solution NMR really is not the method of choice anymore if we want to go to assembled system. We can look at assemblies by another NMR methodology, namely solid-state NMR. And we can make all kinds of different assembled structures in the test tube, rods, spheres, tubes, and so on. And solid-state NMR really is now at the stage where solution NMR was sort of 20 years ago. They can get beautiful spectrum by fast magic angles spinning at high magnetic fields. You can assign all of those spectra, and U means students in Tatiana Polanova's group, who we collaborate on the solid state with, And if you would have shown that spectrum, somebody without telling you that's a solid state spectrum, people would have believed it's a solution NMR spectrum. So we now have complete assignments for the capsid in a tubular assembly by solid state NMR. Solid state NMR is also very good at looking at the dynamical behavior in those assemblies. So you can do a lot of experiments to probe at different timescales. And for instance, here is our tyrosine 145, and we looked at that in particular because obviously it sits at this hinge region and seems to be involved in conformational switching. And we could establish that it exhibits millisecond time still motions in the C-terminal domain as well as the full-length capsid. What was even more interesting to see is if you look at the dynamics of the backbone, and for those of you who don't know NMR, don't worry, we just plot order parameters. If it's a small order parameter, the region of the protein is very mobile. It's around 0.9 up to 1. It's a very rigid part of the protein. If you look at the assembled capsid, you will see that the cyclophilin binding loop, and cyclophilin is a cellular protein that interacts with the capsid, is very mobile. Now, cyclophilin is necessary in certain cell types for the virus to be successfully infectious. So you need cyclophilin around, and it binds to the capsid. If you interrupt that interaction with cyclosporin A, the infectivity is majorly impaired. There are, however, what are called escape mutants, and one of those escape mutants is the A92E mutant. Those escape mutants don't need, they can work without the cyclophilin. And what was interesting to see, if you look at the order parameters for this particular A92 to E92 mutant, you can see that the mobility, which was very, very high in the native wild-type capsid, is now attenuated to a large degree. What was even nicer to see is when we look at the motion in the cyclophilin capsid complex, that is to say, we assemble capsid, we add cyclophilin, again, we see attenuation of motion. And that tells you that it's not always the structural change that can lead to a functional consequence. It can be emotional change that can lead to a functional consequence. And again, NMR really is the method of choice to detect any of those motional changes associated with functional changes. The other method with which you can get structures of assembled entities is cryo-EM. And here I show you two examples of in vitro, in test tube assembled capsid wild type and this one mutant. And then you end up with a reconstruction of your cryo-EM map. And here is one particular tube. You can really see the hexameric arrangement very well on the outside of the tube. This is the parameter I want you to look at, because now you can hopefully remember what the average diameter was of purified capsid cores from virions. That was 44 nanometers. The diameter of this tube is 42 nanometers, so it very much reflects sort of the part of the capsid core that is sort of rod-like. It's a single wall tube, and the sort of thickness of the wall is around 80 angstrom. And if you take the whole capsid protein from top to bottom, it's roughly 80 angstrom. So we have one capsid layer that makes the tube. You then look at the density map, and that's one the tubes come in different helical arrangements. This is a -13 plus 11 arrangement, helical arrangement, and then you fit in your atomic structures. And what was most gratifying to see that when we took our NMR dimeric structure, we could fit it into that density, and we had no clashes, and that was really, really nice. So we did not have to take the solution CTD apart to fit it into the helical assembled structure. And that means that it's probably very likely that the CTD is in the conical capsid the same way as we saw it in solution. And it sort of makes sense. The capsid does assemble from solution conditions. Now, here you see the density, and as I pointed out, the six-fold here, we all knew about because we know from the outside we always see this hexameric arrangement of NTDs. There is the two-fold where the CTDs come together, also no surprise. But we saw density here at this particular area, the threefold. So we looked at this trimer interface in a little bit more detail, and again, it is involved, it's made out of helical interactions. Now it's helix 10 and helix 11. Now the cryo-EM structure is not good enough to see side chains there. However, you know in a helical arrangement what kinds of residues would roughly be here at the threefold. And then we looked in the literature whether what we saw there would make sense. And in particular, we noticed that lysine 2 or 3 mutations were known to interfere with assembly and that they destabilize the capsid. And the glutamine 219 to alanine mutation had reduced infectivity. You can see those are all residues that are at the interface. So that meant that this interface clearly was important for the assembled capsid, and then we made some more directed mutations, and we made a whole lot of number of mutations. I want to show you two here that were sort of maybe a bit counterintuitive. Glutamate 213 was changed to alanine and glutamine, and 213 is sitting here. Now, when you look at the infectivity, this virus is really pretty dead. However, if you look at the stability of the capsid now, you notice that both mutants are more stable than wild type. And what it tells you, in contrast to what you normally would have believed, if you make unstable capsid, obviously infectivity is reduced, and that is true as well. If you make the capsid too stable, you also reduce infectivity. And it means that the virus in coevolution with the host has really found the sweet spot of being stable enough to get into the cytoplasm, but not too stable not to let reverse transcription and then the PIC formation form. So both destabilizing the capsid and stabilizing the capsid leads to reduced infectivity. And what is the most the strongest way you can stabilize an interface? You can make a disulfide bond. So we pick two positions here, change them to cysteine, and as you can see, this one is really, really dead, that virus, and this is very sick virus, doesn't really infect very well. So clamping across that trimer interface really impairs the virus in terms of infectivity and makes the capsid really stable. And this interface we can also cross-link in real HRV particles that we've got from cultured cells. So it's not just in the test tube. So time goes on, cryo-EM methods improved. By now there's a density map at 8.6 angstrom. And now not only do you see just the hexameric arrangement, you can really see individual helices, and you can see the CTD dimer here at the inside of the tube, or here bridging two hexamers. And for those of you who are not used to cryo-EM, I always like to impress people how fast cryo-EM has improved. This is a density map at 16 angstrom resolution, and it sort of blobs. This is the density map of 9 angstrom resolution. And I wish I could put up the 4.5 one yet, but we're still working on that one. Hopefully soon it will be there. Then we really see all the side chain. We then also looked at other mutants now at the interface, and there was one particular one, alanine 204, that we changed to a cysteine. And then when we assemble in the test tube, we can actually assemble in sort of conical arrangements. And this is now something that we will work on to look at the conical structure of the in vitro assembled capsid. And Okay, let me go back here and point out just some of the mutants. And you can see this capsid, really, really stable, but very, very dead. Again, now we have some in vitro assembled capsid, but what we really want to look at what really the conical core looks like. And for those of you who are well versed in geometry, you know that if you have a hexameric lattice, you can never have a closed shell. You have to have pentamers, and enclosed pentamers to make an ovoid. And there is a particular number of pentamers, and these are 12, that you need to close an ovoid. And even Leonardo da Vinci already knew this in the late 1400s. And for some of you, since we're in Germany, you will probably be more reminded of a soccer ball than what Leonardo da Vinci drew there just on geometric means. So all of the people who've made these coarse-grained models knew that you had to put pentamers, and because it looks very nice and symmetric, when you draw it in the test tubes, people would put 7 pentamers on top and 5 at the bottom, so you have a narrower and wider end. But how many are really there, and how do we get there? So what we did there is we made a stubby tube from the cryo-EM and put it all together by using molecular dynamics flexible fitting, a program that comes from Klaus Scholten, to the electron density. So we started out with a structure here in the middle, connected them with a CTD dimers that came from the NMR, and then had a tube as shown here, made out of 71 hexamers, 13 million atoms, conditions as they were assembled in the test tube, which means 1 molar sodium chloride, pH 8, 10 nanoseconds of the flexible fitting, and then 125 nanoseconds MD afterwards. And this was all done on the NSF Blue Waters computer, and in particular, Juan Pereira was a postdoc in Klaus Scholten's lab, did a heroic effort doing that. But then we know we have to add pentamers, right? So we have to make then pentamers that are surrounded by hexamers. And now I have to go and start 2 movies here. These were also done by MD. And for those of you who are in origami, they would, or know about origami, they know immediately that if you make a hexameric lattice and just cut out physically one of those hexamers and then glue back together that seam, you end up with a sort of a curved structure. And that's really why you need to add pentamers into the assembled capsid if you want to close a space. And the color coding on the next slides will always be the same. The CTDs on the inside will be in orange. The hexameric NTDs on the outside will be in blue, and the NTDs will be in green. Now we needed the shape. How do you get a shape of a bona fide HRV conical capsid? You have to do cryo-ET. So you basically go in the cryo-ET and you cut through to get the shape. And then you get the cone angle from this cryo-ET, and then you make Fullerene cone models with different numbers of hexamers, always with 12 pentamers, and check which of the ones fit your cone angle and shape best. And I've shown over here what we actually mapped out in the cryo-ET of the real viral cone compared to a more symmetric Fullerene cone model that has 252 hexamers. And what we found out that for the particular one that we did the cryo-ET on, it fit best a model that had 166 hexamers and 12 pentamers. So where did we put the pentamers? The pentamers we put where we saw the largest inclination when we're going down in the cryo-ET. So wherever we saw an acute angle, that's where we positioned the pentamers. Then the whole system was built up, all atom model again, 216 hexamers, 12 pentamers, everything in a box of water with the correct ions now. That would be physiological ions. The whole system is 62 million atoms, and you can see here now that we have not just the outline, the blobs of the hexamers and the CTD. If you go in there, we really have the atomic model in there in all detail. And trust me, it's now a big, big job to analyze all that in detail and test some of the functional questions that arise from a model like that. How do we get in and out of a cone and so on? Now, with this, I've reached the end. As you're well aware, and hopefully I pointed out repeatedly, this is really a collaboration of people in our center. We really work together as a team. All of the protein we work on comes from Jean Moulin's lab. We make everything N15 labeled, whether it goes to crystallography or cryo-EM. And the CTD structure was solved by Inja Buren with help by a postdoc. All of our functional work is carried out by Chris Aiken's lab in Vanderbilt. He's a virologist, so everything that we structural people think is important gets tested functionally. Pejin Zhang and her group does the cryo-EM. Tatiana Polanova is the solid state person, and her students do all these heroic efforts now assigning solid state spectra. And the all atom model of the real capsid would not have been possible without Klaus Schulten and him having almost sole access to the NSF Blue Waters computer when it got installed first, and Juan, who's been a fantastic person carrying out all this work, really in conjunction with us, looking at the data all the time. And that just leaves me thanking you for your attention and be happy to answer questions.
Yeah, thank you very much, Angela. I just got permission from the main organizer to allow 2 questions, and I see already one person at the microphone there.
Thank you very much. I was just curious, all these structures that you show, they're very static. Is there any evidence that these pentamers diffuse around, so those acute angles kind of change over time?
I believe they do, but I have not any information on that. But I think that's why I'm very, very interested in doing the solid state on those conical arrangements.
Okay, thanks.
Who decides where the pentamers and the hexamers are, and also, I mean, who decides about the size? In principle, you could grow it much larger than it is, I mean, by just having more hexamers than pentamers. So is that, so who decides the size?
The cell decides that. And it happens in the cell, and nobody knows how and why, but it happens.
Okay, so there are still unsolved questions, but maybe better ideas actually come. If you look on the first page of the abstract book, where it says that Springer is celebrating 30 years of EBSA, and there's a free drink, and I heard that this includes champagne as well as alcoholic beverages, and so maybe somebody has a good idea on how the capsid actually is assembled in the cell. Thank you very much, Angela.
There are several generations of young people who has lots of work to do.
Thank you very much, Angela, again for this great plenary talk. And